Welcome to Dr. Reginald Gorczynski's lab. This is Dr. Reginald Gorczynski of Network Immunology. I'm George Hoffman. We're here to chat with Reg today about Abstamex, which is an emerging technology being developed by Network Immunology, Inc. Thanks for being here today, Reg. Very, very much appreciate your time.
Delighted.
Reg has been the one doing the experimental studies for the development of Abstamex as a technology, as a treatment that we're developing towards human clinical trials. Thanks for being here today, Reg. So let's get started.
Okay. Ask away.
Ask away. All right. That's good. Thanks very much. As a person who is both a medical doctor, an MD, and a PhD, and has 45 years of clinical and research experience, you must have the ability to put your time into various projects and ventures, of course. Why is it that you chose to invest your time and money into this Network Immunology, Inc. venture?
Hey, that one's pretty easy to answer. Throughout my whole research career, from '69 onwards, that's nearly 50 years now, all I've ever really been interested in broad strokes, is how the immune system regulates itself and how we can impact on that from the outside to manipulate that immunoregulation. And really this whole therapy, so there was a period, I guess about 25 to 30 years ago, when we ourselves became heavily interested in the whole idea, which built from Journey's original. He's a classical immunologist, for those who don't know. Classical immunologist.
That's Niels
Journey,
yes.
Classical immunologist from the 50s, 60s, who first postulated this whole idea that the immune system can be thought of as a network. So you perturb one specific antigen, one specific perturbation, you perturb this part of the network, and just as we now think about cybernetics, something happens out here, but you had no idea it was going to happen. And that's because the whole thing is really interconnected, and it's a self-regulating system, and it has to be. And the reason it has to be, we know, is because what happens when it doesn't work well. So when the immune regulation doesn't work well, people get disease. We think autoimmune disease, for instance, a classic breakdown in natural immunoregulation. Infectious disease, I don't want to go out too much on a limb, but very often some infectious diseases represent a breakdown in immunoregulation that we can't control. We were built to be able to control infection. You break down immunoregulation, you can't control it. A lot of cancer biology is predicated, the approach to that is predicated on the notion that, again, we should be able to clear malignant cells. They represent, if you like, infections of aberrant cells. The reason we can't break down immunoregulation. So we became very interested in network theory and immunoregulation. And the immunologists, the pure immunologists, developed their own language for this. And they regard the initial reaction, the initial challenge, as producing an antibody against the challenge, which they call an anti-antigen challenge. And then there's a reaction from the body within to regulate that, which produces an antibody against that antibody, that they refer to as an anti-idiotypic antibody. And then it's a third string cybernetics. You get an antibody against that antibody, which is against that, so an anti-antibody, and so on. And it goes on in continuity. And we did some work, I guess, 20 odd years ago, looking at this, and published several articles that said, this really was happening, guys. And there was evidence for anti-idiotypy as a controlling feature in immunological paradigms. And then our work took a slightly different bent, and my lab became very heavily focused on something else, rather more conventional, but a molecule that we thought was involved in immunoregulation in a variety of different spheres, in transplantation, in autoimmune disease, and in malignancy. And then lo and behold, I guess about 2 years ago, Jeff Hoffman, who is the CEO of Network Immunology, contacted me and said, Reg, I just saw this paper that you published on transplantation. I didn't know you were still doing work on immunoregulation? I said, sure I am. Have been for years. Never really diverted from that. And he said, look, he's continued his work on network regulation of the immune system. Would I be interested in trying this approach in one of my model systems? And it intrigued me because, again, it really built on the work that we had sort of left off 20 years ago. But it was a very simple experiment, fit well within our paradigm. So we did the experiment, which involved trying to manipulate a graft rejection response without playing around with the graft rejection response itself, but by trying to manipulate the regulation of the graft rejection response. We did that experiment, and lo and behold, we modulated the graft rejection response. I thought that was very, very exciting. So Jeff said, well, we really need to study this in other models. And he came up with the autoimmune model, and the one he picked right off the bat was diabetes. And you can see why. Big challenge, big problem. So I guess the two arthritis or the two autoimmune models most people would have picked. One would be diabetes. The other one would be joint related arthritis, rheumatoid arthritis, something like that. Pick diabetes. So fast forward 6 months, just to let you know that not everything in science works. Fast forward 6 months, we beat our head against a brick wall there for 6 months, and we really failed to produce a major effect. There was a small effect, tantalizing, intriguing, but really not a major effect. And I said then in a conversation that I think we're looking at the wrong model here. Really, diabetes is very complicated. There's a whole metabolomics that goes into understanding diabetes, and the immune system is just one part of that whole problem. And I think we're not touching on other major issues. So let's get away from that. Let's go to another autoimmune model or another model where we think this is mostly immunoregulated. And we opted then to study another model that my lab had been working on for many years, and that was a colitis model, an inflammatory colitis model. It's a great model for autoimmune disease. We studied exactly the same manipulation that we used in the graft model, and lo and behold, we were able to actually attenuate inflammatory colitis in mice. Very exciting. We were doing very similar work, looking at immunoregulation again in a breast cancer model in mice. I said, let's apply the same therapy there. We did. Lo and behold, we have an effect on breast cancer growth, and more interesting, on breast cancer metastasis. So here we go. We've touched 3 different models. We touched the transplant model, we touched an autoimmune colitis model, we touched the breast cancer model, and this same immunoregulation worked in each one. So now we're very intrigued. I mean, but now, as you're probably aware, everyone says, well, look, we're not really interested in curing mice of diseases. We're really interested in application of those treatments to man. And we thought about that for a while, and I said that I think our fastest approach to bring the kinds of notions and the kinds of therapies we're dealing with to man, our fastest approach to the clinic is going to be via an allergy indication. And here is a disease that not life-threatening. In general, allergies are not life-threatening, like infectious disease, colitis, cancers, transplantation. But they cause tremendous loss of quality of life. It's a huge population incidence, somewhere in the order of 5 up to 8%. And the quality of life impact is enormous. And there aren't very good therapies right now. I mean, you can buy over-the-counter and by prescription chemicals that will attenuate some of the problems you have. So the rhinitis, the runny eyes, the itchiness. You can take drugs daily for that. Allergists and immunologists will argue that we can give you injections to desensitize you to the allergen. And sometimes they work and sometimes they don't work. And I said, so here is a disease entity that's crying out for a different approach. Let's see if this works in that model. And we tried it, and lo and behold, I mean, it worked in that model too. And that's where we are right now. So we've now applied this kind of immunotherapeutic approach to 4 different major disease entities, shown it works in all of them, and we're now pushing forward very rapidly on the approach using as our preclinical model, the allergic disease model, with the hope that we can bring that to human application within a year to 2 years. That's the thing.
Excellent. The technology sounds valid and remarkable. Why is it that nobody else is working in this area, in the area of immune network theory?
That's an interesting question. I think if you were to roll the time back 20 to 30 years, as I said, you would probably have found that there were a lot of people working in this area, studying the ability to manipulate a response against one particular insult, which in immunological terms I'll call an antigen. So a response against an antigen, to regulate that by perturbing the response that the body makes to that response, because that's what we're talking about here. And since that time, really the whole science dynamics has changed, and the big change, I guess, came about with the ability to clone genes. And suddenly everyone adopted a very reductionist approach to scientific endeavor, and people started to ask questions about whether we could clone a gene that controlled the response. Just one gene controlling the response. It's a very reductionist approach, but you could at least now ask that question. And a lot of people set off on that approach, and that's where the field has gone in general. And I must say, I mean, in some respects, it's been quite successful. We've actually managed to clone and understand some of the pathways that are involved in producing a response against an antigen, and we've been able to understand them in much more detail. But if you're trying to imagine a pathway, a series of interacting pathways that control the ultimate end product, trying to clone and understand the cybernetics of how all of those genes interact, that's really damnably tough. I'm not going to say impossible, but I mean, nobody is approaching that right now. And our approach isn't like that either. We take a step back from that even and say, I don't have to understand that in detail as long as I understand in general terms how I can perturb something here and produce an end product here. And that's the approach we've adopted. It's interesting. When I was I'll give you a story. When I was a student, a PhD student, so way back, 1969. So a real, I mean, I can still remember 69. And I was being taught by a clinical pharmacologist, a really brilliant man, Bob Bruce, at the Ontario Cancer Institute. And he was giving a course on chemotherapeutics that were being used in cancer patients at the time. But it was a mechanistic course. And he was trying very hard to get us to understand how these chemicals worked in terms of their effect in chemotherapy. And some of them intercalate in DNA, some of them cross-link DNA. And at the end of the lecture, I remember him looking at the audience and he's saying, but the bottom line is, when the patient has the tumor, they don't really care how it works. They want to know, does it work? And that's the bottom line. I mean, it really is the bottom line. So cloning the genes and doing the great science, I'm not knocking the science, but the real big question is, does something work? And if what we're doing works, I'll worry about the exact mechanism. I can give you the theoretical mechanism in broad strokes, but I'll worry about the exact mechanism later. Your immune system perceives an insult. That's the antigen. Okay, so here's an antigen, and my immune system is now going to react against it, and that's the reaction. It encapsulates that. In simplistic terms, it encapsulates it. So we consider this is an antibody, and it's reacted against that. And my immune system now needs to regulate that. Any reaction can't be allowed to go on unchecked. So it needs to regulate this. So it produces now a second-tier reaction which regulates this. There is the anti-idiotypic reaction. So But this we know then starts to mimic. So here's my antigen. That's the anti-antigen reaction. Here's the anti-idiotype. And simple network theory, it's hard to grasp this until you try to draw it out yourself in a map. But simple network theory says this antibody that's reacting against the antibody that's reacting against the antigen suddenly starts to look an awful lot like the antigen, which was why the body now starts to make another antibody against that. And these whole things sort of link up in this huge network until it all dampens down. Just as if you imagine a spider's web, and it's the analogy everyone always uses, if I tickle the spider's web close to one end, I see a ripple effect. It's the same as if I throw a pebble into a pond. But as the ripple extends outwards, the quantitative perturbation of the web and the perturbation of the waves on the lake gets less and less and less and less, and eventually dies out. And that's what network theory is in the immune system. It regulates up close very tensely to stop this reaction from it getting out of hand. It perturbs a lot of other things because these other perturbations are taking place not just in relation to that initial antigen, but it shouldn't be a surprise to know that as this drifts outwards, the perturbations are actually taking place in parts of a network that primarily aren't dealing with this antigen, but are dealing with another antigen completely. That's the spreading effect, but that spreading effect is dampened as you go out. So that's what immunoregulation is really all about. You're perceiving this huge network which spreads out in all directions, but it's not quantitatively the equivalent in all directions, or as it moves outwards, it dampens. So that's, and we're trying to manipulate this initial reaction, not by regulating that initial reaction at all, but by coming in here.
Fascinating. That's a great description. I get that. Thank you. So could we say that this treatment is not similar to a vaccine?
No, absolutely not. So the vaccine is the antigen. I mean, I'm trying to elicit something right up front. And that's the way most people think about the first order reaction. And very often, chemists will tell you, if you start looking at second order reactions, they're much more difficult to quantitate, they're much more difficult to predict because they are second order. They depend upon the perturbation from the first order. Mm-hmm. You can imagine now how tough it is to predict a third order reaction or a fourth order or a fifth order. And we're actually dealing with third order reactions. We're actually dealing with third order and first order. That's really the way to think about this. It's very tough to predict them. It's certainly not the way most people conventionally try to perturb a system.
Great, perfect. So what would you say fundamentally makes this treatment different from all other existing treatments that exist?
I think the theory behind it is fundamentally different from the way other people are thinking of changing biology or changing a response to a disease. People don't use this kind of theoretical approach to try to manipulate disease. They deal, as I just said, they deal in first order approaches to manipulate disease. And that's not what this is. The advantage of this is that people have always said that what we really want to do in a lot of diseases is use your body to heal itself. Well, your body's big healer in broad strokes, again, is the immune system. And that's what we're trying to do. We're trying to perturb the immune system and set it back into the balance where it's not diseased anymore, if you like. So
you're by fourth and fifth orders, not just first orders,
right?
Are you referring to these antibody interactions?
Right,
so
exactly.
The first order would be antibody interacting with self-antigens, and the second order would be antibodies that interact with antibodies that interact with self-antigens, and third order would be antibodies that interact with antibodies that interact with antibodies that interact with self. And that is, if I'm correct, that is the network understanding. That is the network
That's
theory,
exactly what we're talking
network
about.
theory of the immune system, right? And that is massively, as I understand, is generally a layperson who's picked up a little bit of immunology, but that is massively overlooked. The fact that these antibodies are recognizing each other as a network is a major part. I mean, what baffles me and just entirely in blows my mind away is that why isn't the whole field of immunology is not acknowledging the fact that these antibodies are interacting with each other via network interactions. That seems like it's like trying to understand the internet, right? It's like you look at the internet, see it. What is the internet? The internet is a network of computers. How can we understand it as a network of computers? Does that whole network communicate within itself to itself? And how do the computers, interact with each other, communicate with each other? Well, via these network interactions. Would you say that the same would apply to the immune system?
Very much so. I think clinicians have an advantage in many ways from basic scientists in this regard, because I think clinicians long ago, as part of the practice of medicine, long ago have to accept the fact that they're going to be doing things with people and to people in terms of therapy that you can't really give a complete mechanistic explanation for. It doesn't mean, I go back to that Bob Bruce quote, doesn't mean that it's not efficacious. It means you can't understand it right now. Scientists in general don't like that. They like to be able to explain mechanistically everything that's happening. So for a scientist, living with the unknown is a very uncomfortable feeling. Clinicians have to get past that very early. Which I guess is the closest way to think of it is as a food allergy model. We give the animals something to drink that causes weight loss, diarrhea, and they do develop an allergic response to it. And the classical way we measure allergic responses is by looking at a particular antibody production, IgE antibody. So they develop an antibody response to that. I might say that some people use the same antigen, because we're using ovalbumin fed to the mice. Some people use the same antigen and actually insufflate it so the animals get to sneeze. So it can be used as a respiratory allergen as well. That's not the way we used it. But in every experiment we've done with the animals fed ovalbumin, they develop all of the pathology of an animal who's got a food allergy. So they lose weight, they look sick, they get diarrhea. We can stop that. We can stop that entirely with this treatment. And then the most exciting thing was that we could then continue to give them the allergen. So we continue to expose them to ovalbumin, but we stopped the treatment after 5 doses. And we asked, were they still protected from the ovalbumin allergic induction? And the answer was yes. They were still protected many, many weeks later. And so again, you have to remember the mice live for about 18 to 24 months, so 2 years. Humans live up to, let's say, 80 years. So there's a 40-year lifespan difference. So if you protected mice for 6 to 8 weeks post the last therapy, that translates 40 times 6 to 8 weeks. That translates into nearly 240 to 250 weeks of extra protection, another 5 years. So one short course of therapy for 5 weeks, and they're protected for 5 years. That's a very profound effect. Now, that's a gross extrapolation. Some people might argue you can't do that. You can't just multiply everything by 40. I'm not saying it's a one-on-one correlation. I'm just saying that you have to remember it's not likely to be just 5 or 6 weeks in man, because the projected lifespan of mice is very different. So it was a very profound effect. I was intrigued, and I was very pleasantly surprised that the effect persisted for so long.
Okay, and so we've seen that we are using something other than the allergen itself as a therapy with this treatment, correct? Now, that's one difference. So it's the same therapy, if I'm correct, for all different allergies.
Yeah, you can remember what we're talking about here is immune perturbation, and as I gave you that example of a web spreading out, and I said when you get out to the 4th, 5th order, the perturbations out on the periphery are actually not even primary perturbations in response to this antigen, they're in response to other antigens, and that's what we're dealing with here. We're dealing with applying an antibody and an anti-idiotype antibody to one antigen, but the effect we're seeing is on an immune response to something quite different. So yeah, in principle, the allergen to which you're allergic is irrelevant in the context of the treatment we're using. One treatment sort of fits all.
Excellent. Okay, so it's a universal treatment for allergy.
Yeah, that's certainly the way it would look.
Is it better, and if so, why is it better than, say, something like a Claritin or other antihistamine-based treatment?
Well, the simple response is the answer I already gave you. I mean, I can stop the treatment after 5 weeks, and the mice are protected for at least another 6 weeks beyond that. I should say at least 6 weeks. We didn't go beyond six more weeks before we tested them. If you're going to use pills to treat your allergies, you're taking them every day. And as soon as you stop taking them, the symptoms are going to come back. So you take them every day for the rest of your life, if that's the route you want to go. I'm not knocking that route. Some people are quite happy to do that. But that's what you're locked into. You're going to be taking this medication daily.
Because inflammation is a key driver behind aging, could this potentially be an anti-aging technology?
I'm probably a good person to ask that question because we've actually been very interested in age-related research and aging research for 20 to 25 years. That's been one of my interests as well. That field is probably rife even more than the cancer immunology field, with people who've come up with various. Everyone wants the elixir of youth, although, you know, if you read Dorian Gray, maybe you shouldn't be hankering after it so much. But we would all like, I think ideally, and I gave this lecture many years ago in Turkey, I think ideally what happens, I'm just going to plot a graph with my hand here. If you look at people's health overall, what happens, you start off very healthy, and then starting at around 50, things start to go wrong. And depending on how long you're going to live, things start to go wrong, and various parts of your system, you know, your musculoskeletal system, your heart, your brain, your chest, they all start, and you gradually dwindle. And right now, given Western medicine's success in treating various diseases, we can dwindle on to 80, 85, 90. But the quality of life at the last 5 years is not that great for a lot of people. I think the approach most aging biologists would like to see us take is an approach that isn't designed to improve the quantity of life, because I suspect there is a limit. What that limit is, I don't know. I've heard some people argue it's up around 125 years, but probably not much longer. Some people argue it's 150. But instead of focusing on the upper limit and improving quantity, let's improve quality. Wouldn't it be so much better if we all lived like this and then just went like that? When we die, that's it. We just go from 100% quality of life to 0, but then we die at 0. And that would be much better. If we could improve everybody's quality of life, then quality of life would come naturally. And so again, how do you improve quality of life? And take your pick. Some people would argue it's nutrition. Some people argue that the immune system does tend to falter as we get older. But is that the key? Is that the key that's causing aging? I don't think we know the answer to that. I think there are multiple things that are probably going wrong that are reducing quality, and we need to tackle them all. And the immune system is but one of those.
Perfect. Thank you. And if this treatment works as well in humans as it has been working in the small animal models, should this treatment be then administered through national healthcare systems, such as vaccines have traditionally in the past been administered? Do you think this is something that should be integrated into healthcare systems?
That's a political decision. Every healthcare system depends on, before any healthcare system decides to pay for something, they want proof of efficacy. That's quite different from getting approval for introducing something into the medical pharmacopoeia. Introducing something requires proof of efficacy, but not to the extent that proof of efficacy for the governments to start paying for something requires. So I suspect you're years away from that. And it depends really on how effective and cost effective it is relative to the treatments that I mean, a public healthcare pot is only so large. And we already know how we're spending a lot of that pot. Vaccination, I think, is a very appropriate way to spend that pot. And if the government then says, I'm sorry, there's not a lot of money left over after making sure everyone gets vaccinated and gets basic health care, then so be it. I mean, that's what we can afford. Recently. I think most people who are open-minded and understanding enough will see that this has potential to be quite revolutionary in a variety of fields. And potential investors or other interested scientists would get on board simply because of that. That's all I would say.
Would you think it would be interesting to collaborate with other groups researching things like Alzheimer's disease and that are lately being seen to have a lot to do with inflammation and diabetes?
Yeah, I mean, yeah, absolutely. I mean, there are a variety of fields. I mean, again, if you come back to network theory, people who are interested in the immunological network theory jumped immediately on this whole idea that the immunological network looks a lot like the brain network. Equally complex, equally unfathomable at the present time. Maybe if I was talking to you 200 years from now, I wouldn't be saying that. But right now, very unfathomable, but very intriguing. And is there the possibility that that the same kind of network approach and the same kinds of immunotherapeutics that we're using to regulate the immune system may actually be used to regulate some of the aberrancies that are appearing in the elderly population, as senile dementia, Alzheimer's dementia, a variety of other chronic, debilitating, degenerative CNS diseases? Yeah, I mean, I think that's fascinating. Interesting idea.
Taking a step back now on this note of fascination, why did you become an immunologist? What was it about immunology that had you be interested?
Oh, my one of my professors when I was an undergraduate student in England, Rodney Porter at Oxford. He just moved there from London, and I found his lectures absolutely fascinating. And I had only just recently switched from doing pure chemistry into biochemistry, and I was extremely fortunate in that my tutor had also made the same switch. R.J.P. Williams, brilliant chemist, and he had become, very late in life, he become very intrigued by the application of chemistry to biological systems and decided he wanted to understand something about biochemistry. So he and I mean, he was my tutor though. I mean, he and I were learning biochemistry afresh, and he was absolutely intrigued, as was I, by Rodney Porter's dissection of basic chemical principles to understanding biology, and in this case, the biology of immunoglobulin molecules, for which he subsequently won the Nobel Prize.
And what brought you to meet, how was it that you came to meet Dr. Jeff Hoffman and came in contact with Network Immunology, the company?
Oh yeah, that's very simple. So from England, I did my PhD in Canada. The condition under which I did my PhD in Canada was one that I had a Commonwealth scholarship to be here. And the condition was that after I finished my PhD, I had to go back to England for 2 years. But while I was here doing my PhD, I married a young lady who's still my wife, who was Canadian. We went back to England. She was unhappy being in England for 2 years, wanted to come back to Canada, not unsurprisingly. So we came back to Canada at the end of that time, and I've spent the rest of my research life in Canada. And then, as I said, my whole research life has been focused on immunoregulation, and we started to work on idiotypic, anti-idiotypic responses as part of the interest in network theory from Neo's journey 20 to 30 years ago. Jeff Hoffman was doing exactly the same. I knew of Jeff because the immunological community in Canada wasn't that great. It wasn't huge. I didn't mean great that's not a qualitative analysis. I mean, not big. 20 or 30 years ago, and I met Jeff at a meeting. It was either in Ottawa or Montreal, but it was somewhere east of Toronto anyway. I can remember sitting down over dinner with him, and we were just discussing our experiments. It was a very interesting conversation. I'm sure a lot of people around the table thought we were airy-fairy and off to lunch. But Jeff has continued to work in that area. I already explained. I continue to work in immunoregulation, but somewhat in that area too. And then he made this approach to me 2 years ago.
And Jeff Hoffman has his own view of how the immune system works according to his version of immune network theory.
Mm-hmm.
Do you have your own version of immune network theory that would be somewhat different or the same as
it wouldn't differ in key areas. I would say we both published a book many years ago which discussed this, and in key areas we're sort of soulmates. I think we have much more in common than in difference.
Okay. Are there other people in the world at this time that share yours and Dr. Hoffman's views on the immune system being a network as such?
Yeah, I'm sure. Could I name names? Jeff could probably name names much better than I. But when I talk to my colleagues, my close colleagues that I've held for 40 to 45 years, ever since I've been in research about this, I mean, they enter that discussion, and it's not just the case, well, we'll keep Reg has been on this shtick for 45 years, we'll keep him amused, and we'll say, no, they're intrigued by it, they still find it fun to discuss. So yeah, there are a lot of people who I think are very interested still.
In this network?
Yeah, absolutely.
Very interesting, okay. Are you aware of any other companies or individuals who are developing technologies based on the network understanding of the immune system?
No, that again, no. The answer to that is very simply no. Does that mean that there aren't any? No, it just means that I'm not aware of them. Jeff may be aware of them, but I certainly am not. Doesn't mean they don't exist, though.
Okay. Would you say that the IJ paradox was the major crossroads in the history of immunology, where people, the IJ paradox arose, people who were watching around the mid-'80s, as I understand, after Niels Jerne had won the Nobel Prize, largely for his work on the immune system and largely for his budding, emerging immune network theory. So the Nobel Prize had been won for immune network theory, essentially. Then this paradox arose, and shortly after, the prize was won in 1984, the paradox, IJ, arose in around 1985. Was that the major turning point that steered immunologists? Immunologists looked at the paradox of IJ and said, wow, we can't understand this. The whole thing looks like a network. There's these network interactions, but we don't understand this. Let's go over here and focus on the details of the system, this detailed reductionist approach, as opposed to looking at the bigger picture, the systematic network approach. And that's where they really, according to some people, they really threw out the whole baby with the bathwater, in the sense that they threw out all the data and all the theory, the baby theory, with this central paradox of IJ, which they couldn't understand. Do you see?
Well, the IJ paradox evolved out of a series of experiments that said mice of this genetic composition and this genetic composition produce this effect, and then you map their genetic composition closer and closer, and you say, this is the gene locus, this is the region that determines that difference. And then at the same time, this is at a time when people had started to clone pieces of DNA. So now you knew where it was, so you'd be able to clone this DNA. And the DNA didn't exist. There was no DNA that could be cloned that controlled that IJ region. So that's a real conundrum. And if you step back and think in broad strokes again, and you think in terms of immunoregulation, it's not that difficult to understand, because go back to this scenario, the immunoregulation that's taking place here doesn't all have to be understood in terms of first order and second order. It could be third and fourth order, and what's happening out in the third and fourth order is not necessarily going to be linked genetically or at the DNA level to what's happening here. That would be my explanation for why understanding immunoregulation, understanding networks explains that data much better than people who have looked for pieces of DNA to explain that data. But again, you have to accept, you have to be prepared to live with the unknown, to accept that that's an explanation. And as I said, science doesn't handle that very well. It doesn't like handling the unknown. It wants detail.
There is a plethora of data in immunology. There are mountains of results, experimental results, of experiments, of thousands and thousands of experiments. So incredible amounts of details, data. Is there, aside from immune network theory, is there a theory or framework that really shows how all this data, all these details fit together? Is there one comprehensive theory that rivals immune network theory from your perspective? In understanding and explaining how the immune system actually works, how the immune system actually functions and regulates itself.
If you were to build a computer which could take all of the data we have about the various molecules that we know exist in various pathways that we know exist, producing various molecules along those pathways that we know exist, and ask the computer then, if I perturb this, what will happen down here? The computer may or may not give an answer which is what you observe in practicality. Until such time as you have such a system, I don't think there is a good explanatory system. It is a black box. That is what I mean with being comfortable living with the unknown. There is a large element of unknown in there, but I do know that if I perturb this, I see this effect. I'm happy with that.
Seeing the results rather than the mechanism.
Yeah.
So what would you like? There's a lot of research going on in the area of modeling or like simulations and like bioinformatics.
Right.
So are you aware of or do you think there might be interest to try and like merge your work and the immune network theory with this kind of approach?
Bioinformatics works very well on pathways. As long as you know you have defined all of the interacting pathways that influence the one pathway you're studying. The problem with expanding that now to the body as a whole is let's deal with just the immune system. I don't just have to understand every possible interacting pathway that intersects in the immune system per se, but I know that the immune system is influenced by other systems, other physiological systems within that body. The central nervous system is a classic. We know the central nervous system affects the immune system. Most people wouldn't have accepted that 20 or 30 years ago, but I think that the evidence now is incontrovertible. Do we understand how it does it? No, long way from that. So informatics still has to build that approach. And that's the problem right now. The whole system is too darn complex. What I was trying to say with this analogy with the computer will work well if within that black box you've actually incorporated every pathway and every interacting pathway in that black box. But I'm not sure that we know all the pathways, and we certainly don't know all their interconnections. And until such time as we do, that bioinformatics is doomed to failure. But that doesn't mean, so it may be doomed to failure in understanding how when I do A, B happens. But we shouldn't take the failure to understand the pathways involved to say, well, then that means that when I do A, B doesn't happen. That's the reality. When I do A, B happens. I observe that. You don't want to throw out that result. And that's been a problem in science. It really has been a problem because people can't understand something. Very often they've argued that, well, that it didn't happen. Well, it did happen.